Friends, so now I am back with my next video, and in this video we are going to talk about Krebs cycle or the TCA cycle. So Krebs cycle is the oxidation of the acetyl coenzyme A, which is released during after the lipolysis like from pyruvate, and then the oxidation of acetyl coenzyme A to release CO2 and water and NADH molecule, which then enter the electron transport chain to release energy. So this is the most important pathway for aerobic condition, like when you have enough oxygen available. So then this pathway is important to release energy. So please make sure to like, share and subscribe my channel. So what is cellular respiration? As I have talked about, under aerobic condition, the cell obtain energy from the breakdown of glucose. But the aerobic organism, they oxidize completely the molecules into the CO2 and H2O, unlike the anaerobic condition in which the pyruvate that is produced from glycolysis, it is being reduced to lactate, ethanol and CO2 under anaerobic condition when you require immediate source of energy. But under aerobic oxidation, aerobic condition, it get completely oxidized to release CO2 and water, which provides energy, and this process is termed as cellular respiration. So what is the pyruvate oxidation that occurs under aerobic condition? So under anaerobic condition, the pyruvate get reduced to the to lactate, right? But under the aerobic condition, there is decarboxylation of pyruvate to release acetyl coenzyme A and CO2, and this acetyl coenzyme A get feeds into the TCA cycle. So this conversion is mainly occurred by a complex of enzyme called the pyruvate dehydrogenase complex. As you can see, this is the complex of three different enzymes and it involves different substrate. So here you can see the pyruvate, which is a 3-carbon molecule. It get oxidized to the 2-carbon molecule acetyl coenzyme A and then releases the CO2 molecule. And then this 2-carbon molecule, it just enters the TCA cycle. So here you can see what is the TCA cycle. It is also known as the Krebs cycle after the person who discovered it, the Hans Kreb. And then it involves the oxidation of the acetyl coenzyme A to CO2 and water, and this is the most common oxidative pathway for carbohydrates. So during the need for energy, the requirement for energy, all these carbohydrates and fats, amino acid, they release acetyl coenzyme A, which gets into the TCA cycle to release energy. And it is also important because the intermediates that are formed in this reaction, in this pathway, can also be used for the synthesis of different amino acids, heme and glucose as well. So, and the most important thing is it occur in the mitochondrial matrix near the place of the electron transport chain. So the NADH, FADH which are released at this place get feeds into the electron transport chain to release energy. So the TCA cycle, it starts with the different steps. So one of the step is the condensation of the acetyl coenzyme A with oxaloacetate. So the acetyl coenzyme A, which is released by the oxidation of pyruvate, it condenses with the oxaloacetates to form citrate and releases coenzyme A. So here you can see the acetyl coenzyme A and the oxaloacetate, they combine to form a 6-carbon molecule which is known as citrate, and one molecule of the coenzyme A is released and water is utilized in this reaction. So now we have the citrate, it then isomerized to form the isocitrate. So in this reaction, one water molecule is removed to form the double carbon or cis aconitate, and then by the same enzyme, by the addition of water molecule, the water molecule is now added to different places. So here you can see before it was at the 3rd carbon molecule, now it's at the 4th carbon molecule. So from citrate we form the isocitrate. It's just the arrangement of the hydroxyl group from one carbon to the another. And the next step is the removal of the carbon molecule from this isocitrate to form the alpha ketoglutarate, which is a 5-carbon molecule. So here you can see we have a 6-carbon molecule, and now by the action of isocitrate dehydrogenase, we release one of these CO2 molecule from carbon third, right, from this carbon, and then during this reaction we release NADH from the NAD plus. So this NADPH or NADH which is released, it feeds into the electron transport chain to be used to produce energy. And then by this decarboxylation, one CO2 molecule is released, and what we get is alpha ketoglutarate, which is a 5 carbon molecule. And then again we do the another decarboxylation step, in which there is decarboxylation of the of the ketoglutarate to form the succinyl coenzyme A. So here you can see now we remove another carboxyl group and we release CO2, and during this step the dehydrogenase complex, it releases NADH. So until now we have two NADH which are released from these reactions and which get fits into the TCA cycle, to the electron transport chain cycle. And from this succinyl coenzyme A, we convert this to succinate by the cleavage of the thioester bond of this group with the coenzyme A. And during this step, what we get is GTP, and the coenzyme A is released. So from this 4 carbon molecule, succinyl coenzyme A, we get succinate and we get one molecule of GTP. And then from this succinate, what we get is fumarate. So in this reaction, from the 4 carbon molecule, it's just the arrangement and removal of the H2 group and then forms a double bond. And what we get is this FADH2 from this reaction by the succinate dehydrogenase. And the FADH2 that is released, it can also enter the electron transport chain and releases 1.5 molecule of ATP from 1 molecule of FADH2. And now what we have is fumarate. It just get the hydration of the fumarate to malate. So it's the same carbon molecule, the 4 carbon molecule, but in this case one water is added. So first you can see the OH group and the H group. So it's still the 4 carbon molecule but with the hydroxyl group, and what we get is the malate molecule, which is also a 4 carbon molecule. And then this malate is dehydrogenated to form the oxaloacetate. So here you can see the hydroxyl group is removed, like the H2 is removed, and what we get is again NADH. So in this whole reaction we get 3 molecules of NADH and 1 molecule of FADH for 1 molecule of acetyl coenzyme A. So now what we get is oxaloacetate, which can again combine with the another molecule of the acetyl coenzyme A to form the citrate again, and the cycle continues for whatever kind of molecule you have. Because from one glycolysis we get 2 acetyl coenzyme A, so this pathway happens twice, and in the end we get 6 molecule of NADH, 3 per cycle, and then 2 molecules of FADH2 and 2 molecules of GTP, which all can provide energy. And NADH and FADH2 can enter the electron transport chain to release energy. So in short, what we have talked about, 1 molecule of acetyl coenzyme A enters this cycle and we get 3 molecules, one here during the 2 decarboxylation step, NADH, 1 from malate to oxaloacetate, and then 1 molecule of FADH2 and 1 molecule of GTP. So in short, we get energy which enters into the TCA electron transport chain to release energy. So I hope it's clear, and if you have any doubt, please let me know in the comment section, and please make sure to like, share and subscribe my channel. Thank you.